everybody. My name is Masaaki Ubukata, coming from JEOL Japan, Tokyo. Thank you for giving me again this opportunity. So last time I explained about our AI structure analysis functionalities on the software. So today I will also explain again about our AI structure analysis technologies, and also I bring one more another AI text for the chemical formula recommendations. So I will explain it. And after that, I will explain about our metabolomics applications. Okay, so metabolomics, so of course you guys already know about the metabolomics. So metabolomics is the study of the small chemical substances made in the body during its activities. So I can say a small molecule is a quite good keyword for these applications. And this is just a summarized table between the GC-MS and the elucidation for the metabolomics. And of course, we can use the elucidation for metabolomics, but GC-MS is still very advantageous for the metabolomics because metabolomics should be measured for the small species. So you can see, of course, we can use the GC by GC. So metabolomic samples, I can say this is very complicated, and we can use GC by GC for the highly separation for the metabolomic samples. And as you can see, so TMS derivatization is a golden standard for the GC-MS metabolomics. So today, so I bring also the TMS derivatives for our AI library. So I will explain later on. Okay, and this is GC-MS qualitative analysis workflow briefly. So of course, we have many steps for the GC-MS qualitative analysis. So introductions and the separation for the 1D or 2D GC, and extraction for your unique compounds, and the final phase should be the identifications. And of course, we are using the NIST database search, and you guys also use NIST database search. But if I want to ask you guys, so if your compound is not listed in the NIST database, how to identify the unknowns? Of course, we can read the mass spectrum manually, but it takes a long time, and it needs knowledge to understand the mass spectrum manually. So this begins our motivation to develop our AI technologies. So this is our theme, how to identify the unknown compounds which are not registered in the database by NIST. So again, NIST database is a quite golden standard for the GC-MS qualitative analysis, and it has over 300,000 compounds in database. But actually, we can recognize more than 100 million compounds in the world, especially in the PubChem database. So I can say almost all compounds are not listed in the NIST database. So right now, so we made a predicted EI mass spectrum database by machine learning, deep learning. And this application is coming from our GC high resolution time of flight and AI-based softwares. So our time of flight has over 30,000 mass resolution and 1 ppm mass accuracy. And we have the 4 kinds of ionization, EI, CI, and photo ionization PI, and field ionization FI. So these CI, PI, FI are soft ionization techniques to get the molecular ions. And our software MS500 AI, so it had over the 200 million compounds database for the predicted EI mass spectrum. It is huge database using by machine learning. And the latest version, so we can handle GC data also. So last time I explained this software briefly, and today, so I will explain about, again, about our workflow and AI text. This is our software workflow. This is a little bit complicated, but I will try to explain briefly. So this software can handle 2 kinds of data. One is GC EI, and another one is GC soft ionization data. So if you prefer to use a CI, of course, we can set a CI in here. And if you prefer to use a PI or FI, of course, you can set these soft ionization data. But anyway, so this software can handle 2 kinds of data, EI and the soft ionization data. And the first phase called it integrated analysis. So in this software, of course, at the beginning, we are doing the NIST database search. And also, this software will do the EI fragment ion analysis using the high-level mass spec. And also, this software will do the molecular ion analysis coming from the SI data. And if these results are consistent completely, so we can get the high confidence of the identifications here. But if these results are not consistent, software will go to the next step for the AI structure analysis. And in this phase, so we can do the database search. It is very similar with the NIST database search. So we can do the database search using our AI library. But this AI library has over 200 million compounds in here. It is huge, okay? So in that case, so molecular ion information is very useful to narrow down the structures. So we decided the molecular formula in this data, so we can just narrow down to the isomers from the over 200 million compounds. Then we can get the structures, formula information, and also that we can get the substructure information from the EI mass spectrum, and also we can get the molecular formula information all at once, even though this is unknown compounds. Okay, so actually we have the two AI models for this AI library. So one AI model, so it can predict EI mass spectrum from structures, okay? And also this AI model can predict the retention indices from these structures, okay? So we can get the predicted EI mass spectrum, and we can get the predicted retention indices values, and we put the structures and the spectrum and the retention indices values to the AI libraries. And actually, we provide 200 million structures by ourselves. Then we made over the 200 million EI mass spectrum database with retention indices values. So we have started this project from the 2022. So right now, so our software is version 3. So actually, we improved the AI model version 1 from version 1 to version 3. So this is a diagram of the cosine similarity between the observed and predicted for the 10,000 known compounds. It means that, so we already know the mass spectrum for these 10,000 compounds, and we already know about the structures, these 10,000 compounds, and we compared, measured, and predicted. Then we can get the average cosine similarity value is 0.86. Okay, so this is just an example for the comparison between the measured and the predict. So this mass spectrum is coming from NIST, from these compounds, and this red mass spectrum is coming from our AI model, predicted mass spectrum. So you can see almost all matching between the measured and the predict from our AI model. And this is kind of the averaged values. So just cosine similarity is 0.81, but still we can see main fragment ions are completely matching, okay? So again, we have started this project from the 2022. So version 1 has a 0.72 as cosine similarity averaged values, and the version 2 had 0.8, and the latest version has 0.86. So our AI model is getting better and better, and we still keeping to develop this AI model. So hopefully next time or next 2 times, so I can say 0.9 or something. But anyway, so our model is getting better and better. And this is the actual software's graphic user interface. So again, AI library has a huge mass spectrum. So we have to narrow down the structure candidates. So we can use the molecular formula information, then we can see all isomer structures all at once. So this is just an example. So actually in our AI library, so this kind of the compound isomers had almost 3,300 in our AI library. And we can see all isomers all at once. But actually, we have to check higher cosine similarity scored structures at the beginning. And many customers and researchers asking us, So how about predictions accuracy about using your libraries? So this is just a summarized table between the version 1 to version 3. And first means, so correct structures had highest cosine similarity value on the AI library. And in that case, so actually first candidates should be in here. So actually in using the 10,000 known compounds, 56% compounds are positioned at the first position in here. And in the top 5, so actually this is the top 5 candidate structures. So 82% compounds are in the top 5 area. And in the top 10 in here, and 87%. Okay, so actually, I have to say our AI model is not still perfect, but we can get highly useful structure information was obtained for the 87% of the evaluated 10,000 known compounds. And we provide 200 million structures. So 100 million structures coming from PubChem database, and also another 100 million compounds, we made it in silico. So 27 million compounds for the material polymer pyrrolizate. So we made this kind of the polymer pyrrolizate compounds in silico for the 49 homopolymer and 18 copolymers. And also we made 75 million compounds for metabolomics for the TMS derivatives and the methoxym derivatives by in silico. And we input these over 200 million compounds into our AI model. Then we made AI libraries. Okay, and this is I'm showing again about our AI workflow. So in this workflow, so molecular ion analysis result is quite important for both integrated analysis and AI libraries analysis, because we are using the molecular formula information for the narrow down the candidates. Okay, so this is another workflow, so totally same, but different workflow things. So at the beginning, so of course we have to measure the samples by the GC-TOF, and we can get the EI and the substantial data. And if your compound is not listed in NIST, so we want to do the AI library search. But before we go to the AI library, we have to decide molecular formula. Okay, so in this phase, but sometimes we will get multiple candidates of the chemical formula also. So in that case, researchers have to decide by themselves which one is correct or which one is incorrect. It is a little bit complicated and take a long time to understand the correct information. So this began our another motivation to develop another AI models. Okay, so this is just an example. I think this is the TMS compounds also. So top one is the EI mass spectrum. We cannot see the molecular ion here. And the bottom one is the FI mass spectrum. And we can get molecular ions clearly. And if we do the elemental compositions calculations for these ions, so we got multiple candidates for the chemical formulas. So on this software, we have two filters. One is, of course, isotopic pattern filters, and another filter is the accreted mass of the fragment ions. Okay, so after we use the two filters, still we got 3 candidates. So in that case, so researchers should decide which is correct or which is incorrect. Then we can do the AI library. But sometimes it is very difficult. So we made a new AI model to recommend which one is correct or incorrect. So our AI model can calculate probability distribution of the abundance of each element. So for example, from this mass spectrum, our AI model calculates probability distribution for the carbons in here. So this AI model is saying, so almost 20 is the highest probability from this mass spectrum. And about the nitrogen, so number 1 is the highest probability from this mass spectrum. And the number 2 is 0 probabilities. And the number 3 is medium probabilities, and number 4 is 0, and the number 5 is lowest probabilities. And we calculate, this AI model can calculate all elements from this mass spectrum. Then we can use this result. And the final phase, and this AI model will recommend, so this one is correct, maybe. Then we can go to the AI library search. Okay, so before develop this AI model, so some researchers stopped in this phase. So the researcher cannot decide which one is correct or incorrect. But right now, so we have another AI model to recommend which one is correct or incorrect. Then we can go to the AI library. Nobody stopped at this phase or at this moment. Okay, so from here, I will explain about metabolomics application using our AI text. So today, so I'm talking about shortly and briefly about HeLa cell metabolomics applications. So HeLa cell is one of the human cancers metabolites cells, and this is very known as a model samples for the metabolomics. And actually, this is a collaborative work with Professor Tsugawa, Tokyo University of the Agriculture and Technologies, and he found this compound N-methyl-UNP by himself. And actually, this compound is not listed in the NIST database. So, but he decided these structures by himself using the EI mass spectrum and the MSMS spectrum manually. And this N-methyl-UNP is a very variable compound, I think, because it may help us to find hidden RNA modifications, because it is a UNP compound, and this is a modification at here, and he found. And we tested this compound, so how to identify these compounds using our AI text. Okay, this is our GC GC-TOFMS measurement conditions. So we are using the inside sample modulator coming from Cepsol, and we are using the EI-FI combination ion source, and we are using the time-of-flight mass spectrometer in here. And this is the 1D GC measurement results. So we can see many metabolites, including the amino acid and the phosphate, sugars, organic acids, something like that. And go to the 2D GC. So we measured over the 8,000 compounds in the 2D GC measurement results. And this is enlarged 2D TIC. So you can see this here is N-methyl-UNP's position, and compound A is co-eluded by the 1D GC dimensions. But of course, this is the GC-GC, so we can completely separate from compound A to the N-methyl-UNP. And you can also see the another compound in here, so they have the very similar substructures. So I can say 2D GC is quite helpful to understand the structure also, because they have the almost same polarities, and they have the very similar substructures. And this is a mass spectrum of the N-methyl-UNP, EI, and FI. So in EI, so we cannot see any molecular ion in EI, but FI, so we can see the molecular ion clearly. And of course, this is not listed in the NIST database search. And this is the integrated analysis result for the chemical formulas. So this is a completely matching for the same with the N-methyl-UNP. So this is the time to go to the AI library. This is just a testing. And actually, we got 53 compound candidates in our AI library. This is all same molecular formulas, isomers. And we are very lucky, so because the N-methyl-UNP was in the first positions. Okay, so we have very big confidence for this kind of the real unknown compounds analysis using our AI libraries. So this is my conclusion. So again, so we made over 200 million predicted EM mass spectrum database in our softwares, and 75 million compounds for the TMS database for the metabolomics. So I think it is very useful. And again, it is very important to combine molecular formula information for the integrated analysis and AI library search. Okay, so this combination of these technologies is expected to be useful in the advanced metabolomics, and also that we can use this technology to other GC-MS qualitative analysis. So actually, we have one more oral presentation and 3 poster presentations today and tomorrow. So if you have interest, please stop at our poster sessions also. Okay, so thank you for your listening. Thanks so much.